Biogenic amines. We're talking about hormones. We've done steroids, talked about insulin, did some proteins right there. That's a polypeptide. Now let's look at these smaller hormones that are extremely potent. Now, interestingly, the first three over here, the catecholamines, they're also neurotransmitters, right? So dopamine can be a hormone or it can be a neurotransmitter. So what's the difference, right? And what's interesting is nerves release these both as hormones and neurotransmitters. So that's kind of confusing. Dopamine actually has a couple different names inside the central nervous system, but dopamine is a really important hormone, as is epinephrine. It's these two in the central nervous system that actually work together in certain concentrations in different parts of the brain that when they're off, lead to all of the mental health issues like depression, bipolar disorder, ADHD, those kinds of things are all driven by actually these hormones. And if they're balanced within the central nervous system. So there are dopamine uptake inhibitors. There are ADHD medicines that increase epinephrine in the brain. So that's how most of those drugs work, by increasing or decreasing one or more neurotransmitters. So what's the difference between a hormone and a neurotransmitter? The difference is location. So a neurotransmitter is really local. It's the control of a nerve controlling another tissue cell, all right, or group of cells. So it's very, 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 very localized. The nerve actually delivers the neurotransmitter right to the door, if you will, or right to the membrane of the cell of interest. Where if I'm going to release dopamine as a hormone, I'm going to put it in the blood supply and then it's going to travel somewhere, right? So it's got to travel through circulation to get to its target tissues. And the receptors might be slightly different as far as whether they're an ion channel or whether they are a second messenger system. It really depends. Now, what I want you to notice about all of the ones on the board is they all kind of look alike in the sense that they all have this ring here, a benzene ring, right? Notice all of them do. And catechol actually means benzene, all right? So a catecholamine is a benzene amine, the amine being the ammonia group, right? Or the amino group here that you can see, which is part of the R group. So these dopamine, norepinephrine, and epinephrine are simply modifications to the actual amino acid we call tyrosine. So you change the R group up a little bit, right? So you really just change this part of the molecule up and add maybe a hydroxide over there to the benzene ring, and you get these really active physiological molecules. Now with thyroid hormone, we're going to put two tyrosines together, and then we're going to add some iodine to it. So you can see we're either going to add 4 or 3 iodines to our molecule of thyroid hormone. And T4 is called thyroxine, and it's the bioactive form. It's the really, really bioactive form of thyroid hormone. And then T3 is triiodothyronine, or just T3. And most T3 is converted to T4 when it reaches a tissue. So both are released into circulation as thyroid hormone. Mammals produce a ton of this because it is responsible for basal metabolic rate. So you guys all maintain high body temperatures. That requires consistent burst or release of thyroid hormone all day long in order to maintain that body temperature. And so we produce a lot of it. And it's pretty interesting. It's the only hormone that's stored extracellularly. All right? We'll talk about how that is possible in the thyroid gland. Now, there are other amino acids that are biogenic amines. And probably the second most common one here is melatonin. You guys all heard of melatonin, right? In us, it's what causes us to be sleepy or tired. When it gets dark, you release more melatonin, and supposedly it helps you sleep. In other vertebrates, it's really important in seasonality. So differences in light during the year control the amount of melatonin. So light inhibits melatonin release. And it looks like this. notice it has 2 rings. So again, a very nice 3-dimensional structure, very stable. And that amino acid up there is tryptophan, all right? And so this is a modified tryptophan molecule. The ring is called an diazosyl ring. It's got 2 rings to it. You don't really need the name. Forget chemistry, right? But that's what this is. And again, we just modify what the R group looks like, and you get a very biogenic amine here. It's released by the pineal gland, which is in the brain. So the pineal gland is the gland that releases melatonin into the bloodstream. Now we're going to talk mostly about thyroid hormone, maybe, there we go, and how it works. So the thyroid gland is pretty interesting. The thyroid gland is typically found in the neck of almost all vertebrates. It's found in a couple of other areas, but mostly in the neck. Ours is right on our Adam's apple. It just sits right there on top. Looks like a little butterfly or an H, right? So it's got kind of 2 masses on either side of the underside of the top of the trachea here, right? And it's got a little bridge that crosses here of tissue that's called the isthmus, which is basically just part of the thyroid gland tissue. On the back of the thyroid gland, there are these little knobs that make up a whole nother endocrine gland called the parathyroid gland. But this is, it's kind of hard to see with that image there, but that's like a dissection there on a human that shows the gland itself, right? Cleaned up real nicely there. I'll just leave the lights off, right? So now if you take a little piece of a thyroid gland and you work it up for histology, it looks like this under low power. So it's made up of a bunch of circles. These are balloons, right? We've obviously cut through them in cross section. And the balloon is lined with a cuboidal epithelium. So here's a 100x image. And so it's like the best organ to look for classical cuboidal epithelium. I mean, it's beautiful, typically nice square cells with big nuclei. And the balloon is filled with stuff, right? As you can see, it's not clear, right? It kind of stains this opaque, whatever color stain you use. And that is called a colloid. All right, so a colloid is an extracellular material that just is sitting in the extracellular space. So a colloid, this colloid has a name. You'll see it on one of the slides. It's called thyroglobulin. So it's a big, huge protein. It's a humongous protein that is exocytosed into the balloon and fills up the balloon. Okay? That is going to be our source of thyroid hormone. So you can imagine the most common amino acid in thyroglobulin is tyrosine, right? To basically produce a polypeptide high in tyrosine. And then we can digest it, add some iodine to it. We can do all of that in the balloon, right? There's another really important protein exocytosed into that space. It's an enzyme, and we'll talk about it in a second. But these are called follicles. So these balloons are called thyroid follicles, and the cells are often called follicular cells, or they're called principal cells. Sometimes histologists call them principal cells. And they produce those 2 thyroid hormones, T3 and T4, and we're going to talk about how they do that. Now, interestingly, as I mentioned, all of this stuff, this colloid, this thyroglobulin, is thyroid hormone in its stored form. And at any one time, normally we store like several months of it, 3 or 4 months of it, they say, of thyroid hormone are stored at any one time. Now, the interesting thing about the thyroid gland is it can grow, can it? I mean, and when you have like hyperthyroidism, or they're called goiters, or if you get a cancer or an autoimmune disease that causes hyperthyroidism. And what grows are the balloons. They fill up. They get bigger and bigger and bigger and bigger. The biggest goiters typically happen with iodine deficiencies, which doesn't happen here in the U.S. very often because all of our salt is iodinized. I mean, I guess you can buy a non-iodinized salt. Why somebody would do that, I don't know why. But we get, or you can get it from fish, you can get it from some other sources, but most of it comes from our salts. We don't have any problems with that. Salt's pretty cheap across the world, so you don't see it very much. You might see it in some like Amazonian tribe that's in the middle of the jungle that doesn't have access to salt. Or you see it in thyroid cancers, or you see it in autoimmune diseases where antibodies mimic TSH. All right, so they look like TSH, which is going to be the pituitary hormone that stimulates the thyroid gland. The other thing that you'll see a lot of in the interstitial space, and I always point this out, is blood vessels, right? So these are blood vessels that are found in the triangles between, they call them triangular pieces of tissue. And these really flat cells here are endothelial cells, or the simple squamous epithelium that lines the cardiovascular system. So how do these principal cells work? So let's look at them. So this is a drawing. It's an old drawing. It's actually the one that you typically find when you look it up on Wikipedia. Right? It comes from a good source. The other thing that I added to this was this little picture here, so that you see the chemistry behind how the enzyme works here. That's important. So how do we produce thyroglobulin? See the name out here, right? Follicle colloid is thyroglobulin. And you can see they drew a picture of it here. It's got a lot of tyrosine groups hanging off of it, right? And a lot of benzene rings. Those are tyrosine amino acids. So the first thing this cell does is continuously produce thyroglobulin. How is it produced? Like any protein that's going to be released to the extracellular fluid. It's going to go through the endomembrane system, right? So your messenger RNA is going to come out of the nucleus. It's going to find a ribosome. It's going to have a signal sequence, right? It's going to be shuttled to the ER where translation finishes at the ER, producing a vesicle off of the ER that's filled with thyroglobulin. And then the thyroglobulin will be exocytosed as it walks along microtubules using motor proteins like dynein, and exocytosed continuously into the follicular space. Another enzyme that's produced, and there are, I've read several papers, there are varying degrees on where. I assume it's produced in a different vesicle, is called thyroid peroxidase. Who was that? So thyroid peroxidase, okay? Now, the difference between thyroglobulin and thyroid peroxidase is added to the membrane itself. So it's not sitting in the vesicle cavity, it's actually going to be inserted in the Golgi into the membrane, which means when the vesicle that houses the thyroid peroxidase is exocytosed, it becomes part of the apical membrane. All right, so it's actually embedded, it's an integral protein that is in the membrane of at least most of it. Now there is some of it out in the colloid, I'll be honest with you, at least the evidence suggests that. But most of it, the majority of it is membrane bound or attached to the apical membrane of the follicular cells. And it has a number of jobs. The first thing it does is it is a peroxidase, which is an oxidizer. It removes electrons, right? That's what an oxidizer does. And what it's going to oxidize is iodide, all right? Iodide is I negative, all right? It is an anion. And that's how it comes into the colloid, as a negative ion, which is very non-reactive. All right, so we have to remove that extra electron. And so that's what thyroid peroxidase does. And this is the equation right here, if you really want to know what it looks like. So TPO is thyroid peroxidase. It takes 2 iodides plus hydrogen peroxide plus 2 hydrogens and produces iodine and water, right? And that's what peroxidase does, is it oxidizes and produces water during that oxidation process. It typically uses hydrogen peroxide to do that. And so this oxidation event is our first job of thyroid peroxidase. All right, now how do we get the iodide into the colloid? Well, we're going to collect it from the blood. Almost all the iodine that you bring into your body, like the iodinized salt, is going to end up in the thyroid gland. Anything that's left over, I mean, like over 95% of any iodine that comes into your body is going into the thyroid gland. Any iodine that's left over usually is stored in bone tissue. It becomes part of bone tissue mostly. But the way we get this, the way we suck it out of the blood, is using a sodium iodide symporter. All right, so we're using the sodium concentration. We're letting sodium flow down its concentration gradient to pump iodide into the follicular cells where it's high. And then it escapes through this really cool protein that has a cool name. I like the name. It's called pindrin, all right, which is an anion uniporter that is very specific to iodine, at least here, or iodide, I should say, sorry, here in the follicular cells. And so pindrin allows it to escape into the colloid, where it then is immediately acted upon by thyroid peroxidase and oxidized into iodine. Now, iodine can then be added to the tyrosine groups. All right, can be added to the benzene rings. And thyroid peroxidase does that also, and that's called ionization. And it's random. The enzyme adds sometimes one iodine or two iodines. So it can add one or two. And once you add a couple iodines to either benzene or just one of them to benzene, then the enzyme can do conjugation. The same enzyme, thyroid peroxidase, does oxidation, ionization, and conjugation. And what conjugation is it grabs on to two of the iodinized tyrosines and brings them together and hooks them together. So it's going to bend and fold up the protein so that two benzene rings from two tyrosine amino acids come together, form a covalent bond using oxygen, and you can either put two tyrosine groups that have two iodines apiece, so that would be T4, right? Or you could put one that has two and one that has one together to get T3. And it's random. It's really random. The enzyme just does it randomly. And it spends most of its time doing that. Now, when the follicular cell is stimulated by TSH, thyroid stimulating hormone, from the thyrotrophs of the pituitary gland, it will phagocytize a big clump of conjugated thyroglobulin. And how does it know conjugated versus unconjugated? There are receptors on the surface that recognize attached iodine groups. And so it's going to phagocytize conjugated thyroglobulin into a vesicle, right? This is a phagosome, all right, or a food vacuole. The food vacuole will then undergo proteolysis. So what does that mean? What organelle is going to fuse with that to cause proteolysis? Who's going to make me proud this morning? Which organelle will fuse with the phagosome so that proteolysis happens, which is protein digestion? This is Final Jeopardy question. Oh. No guesses? Oh, don't be bashful, even if you're wrong. So what organelle is responsible for breaking stuff down? Starts with an L. Lysosomes, that's right. So lysosomes are going to fuse with your phagosome and then digest the thyroglobulin into pieces of protein that are useless and then into your two thyroid hormones, thyroxine and triiodothyronine, or T4 and T3. Those are released into the blood. Now, in the blood, they have to be carried by a carrier protein, okay? So what I like about this figure is I like how they organize it. So you got the three major steps, right? You got the production, you got iodine accumulation, you got release, right? Or phagocytosis. So I do like that about it, but it doesn't give you any of the story, right? There's no words on it. I'm famous for that, right? Giving you a picture and just making you listen. But, you know, I'm not mean, so I give you this next figure, maybe. That's got all the steps, right? That's actually out of a different textbook. Now you have the steps, right? In case you were sleeping during this last 25 minutes. Now, when T3 and T4 enter the bloodstream up here, so if you're looking at this figure just to organize you, here's the lumen of a follicle, right? It's filled with colloid thyroglobulin. And then here's a blood vessel that typically runs on the basal side, right, of our principal cell. So here are a couple principal cells or follicular cells. And they're going to use, again, here is your sodium iodide symporter. Here's your pendrin. Here's thyroid peroxidase, right? Adds iodines. So, and when you add one iodine, it's called monoiodothyronine. When you add two iodides, it's called DIT, which is diiodotyrosine. And then T3 and T4, right? Now, once T3 and T4 are released into the blood, they have to be carried by something, right? And there are two proteins that do that. Well, there are lots of proteins. I'm just going to give you two. One of them is called albumin. You guys all heard of albumin before, right? The liver produces that. Remember, it's really important in capillary exchange, right? Drawing fluid back into blood vessels, like we talked about in the kidneys. We call it colloid osmotic pressure. Remember that? That's on the test. Albumin is also a really important carrier protein. It carries things that are fatty, right? Things that are hydrophobic. T3 and T4 are very hydrophobic because they got big rings, carbon rings. The other protein is called thyroxine binding globulin. All right, so it's called TBG, thyroxine binding globulin, which is another liver protein that is specific to just carrying thyroid hormone. So both of those, there are a couple others, but that's enough. I don't have to give you every one of them. Okay, questions on thyroid production? So as I mentioned before, you release bursts of thyroid all day long. That's what maintains your high metabolic rate. If you're a reptile, you don't do this, right? This is why you don't have to feed a snake 3 meals a day. You feed it once a week, once a month, right? And you as a mammal, you have to eat all the time because you need energy reserve or a resource to make ATP, which is driven by thyroid hormone. And so in mammals, you have this nice negative feedback mechanism that's based on the controlled condition of thyroid concentration, right? There should always be a consistent level of thyroid hormone, and I'll be honest with you, I don't know what it is in humans. I don't remember. You could look it up. But if it drops below that level, right, you're going to get stimulation of TSH release. And so basically, the hypothalamus is monitoring that, right? Because remember, I told you it's a pretty neat part of the brain. It's in control of a lot of homeostasis. And the reason for that is because the nerves there actually have some direct contact with the blood supply there. It's one of the few places in the brain where the blood-brain barrier is kind of weak, right? And it allows those nerves to actually monitor what's going on in the blood. And so some nerves up here in the hypothalamus are monitoring all day long thyroid hormone concentrations. And when they drop, they sense that, and they will release this hormone, TRH, all right, which is thyrotropin releasing hormone, all right, TRH, thyrotropin releasing hormone. And as I mentioned last time, the hypothalamus produces releasing hormones, which will have T and R in them. They also sometimes release inhibiting hormones, right? But in this instance, they're releasing thyrotropin releasing hormone, which enters the first capillary bed, which is on the stalk, travels through the portal vessel, and enters the anterior pituitary and stimulates thyrotrophs, right? The thyrotrophs will then release a burst of TSH, and TSH will then travel to its target, which is the thyroid glands, the follicular cells in particular. And they will increase the phagocytosis and release of T3 and T4. They also increase protein production in those cells so that they can replace the thyroid globulin they use and the thyroid peroxidase, which is being used up also. The T3 and T4 enter the bloodstream, and when they get back to normal levels, then the T3 and T4 concentrations inhibit the thyrotrophs and inhibit the pituitary. So once you get a burst, you're going to get inhibition, right? And that inhibition will happen for a while until the thyroid hormone drops, and then you get another burst, right? And then you get inhibition, and then you get another burst. And so you get these bursts of TSH throughout the day that keep our thyroid hormone close to its consistent concentration within the blood supply. Now, thyroid hormone is not only important in basal metabolic rate, but it's incredibly important in nervous system development in mammals. So, and some other developmental steps. And this is why if you go to the doctor and you're like, I want to have a baby, they immediately will put you on folate, right? Or they suggest you take folic acid. All right, folate is high in tyrosine. So you make plenty of thyroid hormone, because pregnancy causes the release of a lot of it, because you need your metabolism up, because you're not only eating for you, but you're helping baby along. Also, that stimulates central nervous system development, particularly, which starts early in babies, and also some of the other organs. And that's why you take folate. And it's also really important in the development of other vertebrates, particularly those that have indirect development. Thyroid hormone in vertebrates that have indirect development, that have like a larval stage, like frogs, it's thyroid hormone that causes metamorphosis. Right? So for them to go from a tadpole to a frog, they have to develop legs, lungs, their eyes move, their mouth moves. I mean, it's pretty incredible transformation. And in some frogs, that happens relatively quickly, like 1.5 months. I mean, that's fast. It could take years in the frogs up here. So a bullfrog, for example, if in northern Indiana, might take 3 years, right, to get to the point where you metamorph. And then it takes, like several months for metamorphosis, or 3 months for metamorphosis to happen, depending on temperature. For my dart frogs, metamorphosis happens typically within about, once the back legs are pretty developed and the front legs start popping out, I mean, it's like 14 days, 15 days, something like that, if the temperature's right. And it's all controlled by thyroid hormone. And what thyroid hormone does is cause mass mitosis and differentiation of cells, right? And it coordinates the migration and production of the axis of the limbs and the changing in the skull. It's a pretty incredible transformation. And so if you're looking at this, this is a figure for some of the research that's been done on frogs. It's been done a lot. You can see that pretty much during growth phases of the tadpole, as they go from small to big tadpoles, there's pretty low TRH, right? This is thyroid releasing hormone. And as you get to a certain size, that picks up, and it's going to pick up over the final climax of growth until it peaks, right? And that is really the climax or start of where you're on the other side of metamorphosis. So you can see that peak happens typically in frogs right when the front legs are about to pop. And then it drops slowly as metamorphosis finishes. And if you were to follow thyroid hormone here, it would do the same thing. It would go up and drop, right? And so it's that spike or peak of thyroid hormone that causes this differentiation. Now, in frogs, it is a little different. The TRH exists, but there's another hormone that's really important here that's released from the hypothalamus. It's called CRH. C is corticotropin releasing hormone. Both thyrotropin and corticotropin releasing hormone can cause TSH release, which increases T3 and T4. This one also controls ACTH release, adrenocorticotropin hormone, which controls the adrenal glands, which are also involved in developmental process in frogs. But we're focusing on just thyroid hormone here. Now, another thing that has to happen in the tissues in order for these animals to actually to respond effectively to differentiation and development of the limbs is you have to express this enzyme in the target tissues, type 2 deiodinase. And type 2 deiodinase, what it does is it takes T4 and converts it into T3. All right, so T3 is, and I think I said this wrong before, T3 is the more potent form of thyroid hormone. I think I said T4 before, didn't I? I think I did. So T3 is the more potent form here. And so all thyroxine, T4, is converted to T3 at the level of the tissue using this enzyme. There's also other deiodinases. There's type 3, which will then take 3 and convert it to 2. And then there's type 4, which will convert 2 back down to tyrosine if you wanted to break thyroid hormone down. So these deiodinases remove iodine, right? Type 2 being the most important one in the tissues that we're trying to control limb development in, because they take all the T4, which is produced and converted into T3, which is much more potent in its effectiveness physiologically. Make sense? Okay, we talked a lot about negative feedback, just throwing that up there again to make sure you understand and kind of understand it in terms of the endocrine system. And negative feedback happens a lot with the pituitary hormones. So remember, the pituitary gland is the master gland in the sense that it often controls other endocrine glands. And so you always have kind of this cyclic setup here where you have, hormone 1 and hormone 2. And hormone 1 here is coming from the anterior pituitary, like TSH, and controlling the thyroid gland, right? And then the thyroid gland produces the second hormone, and when it builds up in the blood, will shut off the pituitary, right? And so it's the concentration of 2 which really controls the production of it, right? It's the actual, typically at least the hypothalamus, but often it will not only negative feedback the hypothalamus, but it'll also negative feedback the target tissue or the anterior pituitary, depending. And so here's an example very similar to thyroid hormone. So I just mentioned ACTH, didn't I? So that is released from the hypothalamus from corticotrophs, if you remember. And they will release a burst of this. The ACTH is going to be released from corticotrophs from the pituitary gland, and will go out and control the adrenal glands, right? So here's your anterior pituitary controlling another endocrine gland. All right, and in this instance, the corticotroph will release ACTH into the bloodstream. It'll travel to the adrenal glands and cause the release of your second hormone here, which is called cortisol. All right, so cortisol is a hormone important in metabolism and in stress. And when cortisol is high in the blood, what it's going to do is just notice it have negative feedback on the pituitary and on the hypothalamus, preventing it from releasing more CRH corticotropin releasing hormone. And as long as cortisol is high in the blood, you're going to prevent the pituitary from producing more ACTH. Well, as cortisol drops and drops to a level below normal conditions, then you will re-stimulate the hypothalamus, right, to cause the release of CRH and another burst of ACTH. And so in this instance, ACTH is hormone 1 and cortisol is hormone 2, right, in this setup. So a lot of anterior pituitary hormones are released in bursts, and then there's a whole bunch of nothing happening, and then a burst, and then a whole bunch of nothing happening, and a burst, because a burst of whatever the anterior pituitary releases causes an increase in some hormone, right, out there, that then shuts down the stimulation of that particular gland that the pituitary is responsible for controlling. Make sense? All right. Here's a couple better pictures of that, showing you typical negative feedback. I love this picture from another text. It kind of gives you the, I always like the nice figures that have the kind of the wording on there for you. Showing you, here's the locus of nerves. This locus right here is the nerves responsible for monitoring cortisol concentrations. They can also respond, believe it or not, to cytokines, which are involved with the immune system, right? And if cortisol is involved with stress and inflammation, it makes sense that if the immune system releases a bunch of cytokines in response to an infection, you might increase your cortisol release, right? But it shows you ACTH release here, adrenal gland control, glucocorticoids. These are cortisols that can affect glucose levels in the blood. And so that's kind of your negative feedback setup there. Showing you the red parts are the parts that actually shut the whole thing off. All right, the last two things I got to really cover for this are just what we're coming to understand is that hormones don't work by themselves. They often work in groups, and they can antagonize each other, that is, do opposite things like glucagon and insulin, right? Where insulin lowers blood glucose and glucagon increases blood glucose. And you can use those hormones to tweak glucose concentration, right? Or they can actually work together, right? And when they work together, they bring about elevated effects of some physiologic response. All right, and this is called synergism, all right? And so when they are synergizing, they often are going to produce an effect that's much larger than the hormone by itself, all right? So here are three hormones, epinephrine, glucagon, and cortisol. All of them by themselves change glucose concentrations. They elevate it a little bit. All right, and notice, by themselves, epinephrine is probably the best one, right? You can see on this graph. So you got hours on the x-axis, and then you got milligrams per deciliter of blood glucose on the y-axis. And you can see that cortisol is pretty bad at causing glucose release, but it does increase glucose a little bit because it really doesn't control glucose release from the liver. It's more about changing the way you make ATP. That is, moving away from glucose, actually, and using fatty acids, for example. We know glucagon does, right? So glucagon, notice it causes an immediate spike in glucose because that's what it does. It causes glycogenolysis, right? The breakdown of glycogen from the liver. But it falls off relatively quickly. And then epinephrine, when it's released, has a nice effect on increasing glucose concentrations in the blood. Now notice what happens when you combine them, though. When you combine them, you get effects that are much larger than just adding them up. Like if I just added epinephrine and glucagon together, it wouldn't equal this one right here, right? Well, look at these. If you add these three together, it definitely doesn't equal that, right? That huge jump. So when all three are released at the same time, your body liberates a ton of glucose. That's synergism, Okay? Now, what's permissiveness? We know what antagonists are. So permissiveness is on the next slide here. Permissiveness is when you have two hormones. One has a general effect on a physiologic response, and one does not. So when you look at these, thyroid hormone does absolutely nothing to fatty acid release. I mean, if you release thyroid hormone into the blood, you don't get fatty acids released. It has nothing to do with fatty acid release. Epinephrine does. Epinephrine increases it just a little bit. Now, when you combine the two, though, you get this huge response. And it's not that thyroid hormone's helping with fatty acid release. It is basically helping the cells notice fatty acids. All right? So thyroid hormone is not working directly on your adipose tissue. It helps epinephrine be recognized by the adipose tissue. So how does it do that? What thyroid hormone does is cause an increase in epinephrine receptors on the surface of adipocytes. And so epinephrine by itself, these adipocytes might have a couple receptors and you get a little bit of fatty acid release. But when thyroid hormone's released in tandem with epinephrine, thyroid hormone goes to the adipose cells and says, hey, add more epinephrine receptors to your surface, and they add more epinephrine, and they break down more fatty acids. So it's kind of an indirect effect of one hormone on another, not that both hormones by themselves cause fatty acid release. Does that make sense? So permissiveness is helping a hormone do its job without affecting the process directly. All right, questions? You guys know about antagonist hormones. I give you one example here. We already talked about insulin, and we already talked about glucagon, but here's another one. We talked about renin, didn't we? And how it causes the increase in angiotensin II, which is going to increase blood volume and increase blood pressure, right, overall. There's an antagonist to angiotensin II, to the angiotensin II system, and it's called atrial natriuretic peptide, or ANP, and it's released from the atrium. You wouldn't have guessed the heart is also an endocrine gland, would you? Right? You would just think, oh, it just pumps blood. But the atria will release AMP when they're stretched too much, particularly the right atrium, which is receiving all of the venous blood from circulation. If a bunch of venous blood is coming back too fast and you stretch the atrium further than normal, that means blood pressure is too high, right? Too much blood is returning and causing stretch. So it releases AMP, which is all the opposite of what renin, angiotensin 1 and 2 do. All right, which would cause more urine production, right? Drop your blood volume and relieve the increased stretch to the heart. There are other examples, but that's good enough. Got enough to know, right? The